This video will focus on Punnett squares. What are we trying to demonstrate with Punnett squares? We're trying to show fertilization happening. So let's say that we start with two different plants. The first plant is homozygous dominant for the big A allele, whereas the second plant is homozygous recessive for the little a allele. We represent that as little a here, big A here. If we perform a Punnett square and show these initial parents breeding, when we cross them, we'll produce the F1 generation here. How would we perform that? All we do to complete the Punnett square is go across and down, cross, down, cross, down, cross, down. By crossing these two individuals, we can see that all of the F1 come out to be big A, little a, or heterozygous. Because this is dominant recessive allelic interaction, these individuals are carrying a big A allele, which means that all of the F1 phenotype will be purple. If we take two F1s, so cross a big A, little a with another big A, little a heterozygous individual, will then produce the F2 generation. Again, we complete the Punnett square by going across and down, cross and down, cross and down, and across and down. What Mendel found in his monohybrid crosses is that in the F2 generation, there would be a 3-to-1 ratio in the phenotype, meaning 3 out of the 4 come out to be purple, and 1 out of the 4 come out to be white. If we look closer at the genotypes, though, we can see that 1 out of the 4 are homozygous dominant, 2 out of the 4 are heterozygous, big A, little a, and then 1 out of the 4 are little a homozygous recessive. But again, the 3-to-1 ratio that Mendel could see and describe was in regards to flower color. So 3 out of the 4 come out to be purple, and 1 out of the 4 come out to be white. What this demonstrates is Mendel's law, how they reflect the rules of probability. So a Punnett square, we have to remember, are representing what happens in fertilization when the eggs and sperm come together. In this case, we're showing two F1s. So an F1 would be big B, little b, and another F1 would be big B, little b, and we're crossing them to produce the F2 generation, which is what we see demonstrated here. This is saying that if these individuals, if one is big B, little b, what that means is half of the time, this individual, when it goes through meiosis, half of the time they are producing big B alleles, and half of the time they're producing little b recessive alleles in their gametes. So in this case, the eggs. The sperm is doing the same thing, right? Half the time the sperm is carrying a big B, and half the time the sperm is carrying a little b allele. Okay? So it's completely by chance then, as far as how those gametes will come together to form the offspring, which are represented here. Okay? In the case of a heterozygote that's carrying two different alleles, it's a 50-50 chance, or half and half chance. Right? So let's say by chance this individual contributed a big B dominant allele, and in this case, this male also contributed a big B dominant allele, right? There would be a 1/4 probability that the offspring in this case would be big B, little b in their genotype. There's also a chance that the individual could contribute a little b from the female egg and a little b from the male sperm, and so there would be a 1/4 probability that the offspring come out to be homozygous recessive, little b, little b. The same idea here and here. In each case, there's a 1/4 chance of this happening, but we could combine those two together and say, well, overall, the total probability is there's a 1/4 chance that the offspring would be heterozygous, big B, little b. To help clarify this concept of what Punnett squares are trying to help us visualize is how are the different ways that sperm and egg can come together. Through meiosis, so cell reproduction, the male is going to produce 4 sperm cells every time, and the female is going to produce 4 egg cells every time. It's completely by chance which sperm gets to fertilize this egg. So it could be by chance that this one fertilizes this one fertilizes that one, this one fertilizes that one, or maybe it's that one that gets to fertilize this one. It could be, though, that maybe this sperm fertilizes that egg, or maybe it's that one, or maybe it's that one. Or maybe during fertilization, it's not the sperm who gets to participate in fertilization. Maybe it's this one, and it fertilizes this egg, or maybe this egg, or maybe that egg, or maybe that egg. Maybe it's not that sperm. Maybe it's the third one right here that gets to fertilize the egg. Maybe it's this sperm and this egg that come together, or maybe it's this sperm and that egg, or maybe that egg, this sperm and that egg. Maybe it's not that sperm that gets to participate in fertilization. Maybe it's this last one. Maybe it fertilizes this egg. Maybe it fertilizes that one. Maybe it fertilizes this one, or maybe by chance it fertilizes this one. Right? You see how quickly this can get very messy. Right? Which one is it? Right? It's much easier to sit here and combine this into a more simplified, nice and neat Punnett square, like what we see here. It's much more easier to visualize the sperm and egg coming together in a much more uniform, clean representation. But it is showing essentially this happening right here. It's just a much easier way to view it and to look at those probabilities. Remember that Mendel, when he did these experiments, he performed them on many different traits. We've been focusing a lot on flower color, such as purple flowers crossing with white flowers. And what do we start with in the P generation? What do we get in the F1? What do we get in the F2? But remember, Mendel was looking at many different examples besides flower color, right? And still got almost the same proportion every time. So now what you can do is practice on your own. So perform your own genetic monohybrid cross between two individuals of opposite traits. Let's say that the opposite traits are green pod versus a yellow pod, such as what we see right here. Mm. Green pods, we're going to use big G as the dominant allele, and yellow pods, we're going to use little g to represent the recessive alleles. So I want you to work through these 5 questions. What are the parents' genotype? What are the resulting F1 offspring homozygous, or are they heterozygous, or are they a mixture of both? What are the chances or the probability that the F1 offspring are yellow? List all of the possible genotype proportions or the ratios that you would expect to see in the F1 offspring. And then what are the chances that the F2 offspring come out to be yellow? So I want you to play with that and see what you come up with.